Biomedical Engineering at the University of Connecticut. His research is highly interdisciplinary and is at the interface of biomedicine, materials, and micronanotechnology. His research focuses on the science and technology to transform biodegradable and biocompatible materials, such as surgical suture polymers and amino acids, into special forms, shapes, or structures with smart functions at micro and nanoscales for diverse applications in vaccine drug delivery, regenerative engineering, and electrical implants. He recently developed a platform technology, so-called SEAL, stamped assembly of polymer layers, to create three-dimensional microstructures of biodegradable polymers, and advanced SEAL method to create a novel single administration of cell-boosting skin microneedle patches. His works have been published in very prestigious journals, including Science, Science Translation Medicine, Nature Nanotechnology, Nature Communication, Advanced Materials, etc. And is also highlighted in major media such as the New York Times, BBC News, The Guardian, NH Research Matter, and others. He's won many awards for his work, including the Young Investigator Award from the Journal of Biomaterials, ACEL Young Investigator Award, MIT Top Innovator Under 35 for Asia Pacific, NIH Trailblazer Award for Young and Early Investigators, and many others. The floor is yours, sir.
Thank you. Thank you so much for the nice introduction. And it's my pleasure and honor to present this talk for Terasaki Institute. I know Ali for a while and really admired him for what he's done. And so this is a great chance to have more interaction with his institute. So the talk today is mainly about the journey that I went through to use this smart biodegradable polymers for a lot of different applications in medical and healthcare field. Before the talk, I need to declare some conflict of interest from two of the company that I founded. The status as a procedure. So basically, what I'm going to talk about is center around these materials. The materials that it used for medical surgical suture, and so it's biodegradable and it's safe at the same time. And so the key question that my lab is trying to ask is that can we just transform these materials into special forms, shapes, structures, and even enable the material to have some exciting electroactive properties so that we can use it for a lot of different medical implant applications and even more than that. And so what I think I'm going to talk today is to describe to you a couple of research. Most of them are published surrounding these materials, and that including the research for drug vaccine delivery, research for tissue regenerative engineering, and then the use of this material also to make exciting biodegradable electrical or mechanical active device like ultrasound. And so to start, I'm going to talk about the first project that my lab is focused on to develop a so-called single administration and cell boosting vaccines or other therapeutics. So the talk is pretty long, so please just interrupt me any time if you want to clarify or ask questions. I feel that would be probably more effective in terms of catching the flow of the talk. And so this work came from the problem that is very common to the traditional way that we vaccinate people or deliver drugs into the patients. And so most of the vaccines include different injections, and that's spaced like every month or even every 6 months or a year apart from each other. And so those injections are very problematic because, as you know, a lot of people, especially pediatric patients, they are needle phobic. They really hate to get injection. And then at the same time, you produce a lot of this biohazard waste, after the injections. And for people who live far away from the medical centers, the transportation of this vaccine to those areas also problematic. And especially you need a cold chain to store the vaccine and make it stable for a long-term use. And so all of that, giving this effort to vaccinate people in a large scale becomes really challenging. But at the same time, everyone knows that the booster shot or the follow-up shot in vaccination is very important. Without the booster shot, the antibody in the blood actually come down significantly just few months. For example, like in this graph of the influenza vaccine published in Science in 2018. And so when I started my postdoc at MIT, working with Bob Langer, one of the projects that we got funded from Gates Foundation is to develop this single injection and multiple pulse vaccine delivery. So the key to do that is that you would have to make this kind of core cell 3D core cell microparticles. The shell would be biodegradable polymer made out of the surgical suture materials. And sorry, that's the shell. And then the core would be the vaccine that you want to deliver. So if you can put 3 of these particles into a syringe, and each of them release the vaccine at different times, then through one single injection, you can get a 3 different release bursts to mimic a 3 different injection over a long period of time. So that's the way we make these single injection vaccines. And so I developed this technology, which I name it SEAL, stand for stem assembly polymer layer. This came from my background as in microelectronics. And so this core cell microparticles, it works really nice, give us the release kinetics that we wanted. But when I use it and sitting in the lab and inject it into the animals, I found so many problems from the microparticles. First of all, it's still very big. It hundreds of microns. And so I have to load each of this into the tip of the normal needles in a sequence. And so that, for me, I feel is really impractical. And so at the time, I started to read about microneedles and the way that the microneedle patch can avoid all of these injections. And that is when I started to think about a different project in which, what if we can just replace all of that injections, right? So injection free, and still be able to make such a core cell structure in the form of this sharp microneedle, and so that we can insert it into the skin of patients. And each of these microneedles can deliver the vaccine in the same way that my previous core cell microparticles still can do. And so with this, we developed a fabrication process at UConn, so that we can make the core cell micro needles, as I show you in the previous slide. Let me see if we can plot it. So this is just a video to show you the way that we make it. So we started with a PLGA film. We have a PDMS mold. We press this on the PLGA, and that form the needle shell. And then we do the impinging to form the core. And then we align the micro-molded vaccine of the drugs with the mold. And then we just insert it into the shell. We spin water and solvent to remove the scum layer, and then we put another layer on it. So this is a layer by layer process, somewhat similar to the 3D printing. But I adapt this technology from the mask aligning process that we usually do in microelectronic fabrication to make computer chip. And then from that, we can insert the microneedle into the skin. Between the microneedles and the backing layer, there's a sacrificial polymer that is super hydrophilic, allowing the microneedles to be released when the patch is in contact with the body fluid. And so with that, we can create this wonderful core cell microneedles. As you can see on the screen, the core is stained by red color. And this is a patch that we have it on this holding in my student hand. And so when you embed it into the skin of animals like this rat, the skin, because the holes that we poke into the skin was really small, we can see that after like 40 to 1 hours, 40 minutes to 1 hour, the skin will completely heal and it encapsulate the microneedles inside this dermal layer, inside the dermis. And that actually really good for vaccine because the superficial or the top layer of the skin is super sensitive to, it has a really sensitive immune cells like dendritic Langerhans cells that it can boost the vaccine efficacies. And so from that, we actually went ahead and loaded our microneedle patch with this clinical vaccine, so-called Prevnar 13. And this is a commercial vaccine that made by Pfizer. So all we do is we just put it into our microneedles in a dry form, and then we apply the microneedle at the time zero, at the day zero. So the microneedle has all built-in booster release, and it programmed to automatically release the vaccine at day 0, day 9, and day 44. And at the same time, we do the traditional sub-Q injection at the same time with matching dose to compare the effectiveness. And you can see that from the single time application of the microneedles on the skin, we get the immune response very similar to the multiple injection over a period of 2 months. But in terms of the immune protections, we can protect all of the animals from a lethal challenge of the bacteria. In this case, it's a pneumococcal bacteria, which is a very dangerous bacteria to cause the pneumonia in the pediatric or maybe old people. And so with this, we're showing the concept of the single administration and cell boosting microneedles, which can replace all of the injection in the traditional vaccination process. But not only at this stop at the vaccine, we actually developed this for a lot of different types of vaccine and other therapeutics. And given the time limit here, I'm just going through all of the project is basically it has happened and also ongoing in my lab. We developed this single time microneedles for mRNA vaccine. We had developed a way to stabilize the mRNA vaccine in such a long period of time, like 6 months. So this is unpublished data, but we also want to develop this for mRNA vaccine. And then we have worked with the SARS-CoV-2 protein vaccine against the COVID-19 viruses. And this is the work that we already published. We also work with antibody delivery. And in this case, we have to maximize the loading capacity of our microneedles. And so we was able to make like 2 milligram per 1 microneedle patch, 1 by 1 centimeter square microneedle patch, and deliver the HIV, the anti-HIV antibody repeatedly over a period of time to sustain this antibody level in the plasma such that we can, sustain the immune protections of the patient against the virus, HIV virus. And then we also use the microneedles to deliver some other drugs like this GLP drug, the semaglutide, a product of Novo Nordisk that we can repeatedly deliver this drug over a period of 1 month, so that instead of getting like 4 injections every week for this drug, for this diabetic or obesity drug, now the patient only need to receive 1 patch at the day 0. So all of this is showing you the ability to engineer, to use our microneedle platform for delivering not only vaccine but other therapeutics. And the key things here is that we can replace all of this injection by this almost painless and skin application of the microneedle patch. So for that, I'm going to move to the next project that we're working on the lab. So what I've been talking about is sort of like transform the surgical suture polymer in a 3D structure to create a novel vaccine or drug delivery system. Now we're going to tell you another part of it is that we're going to transform this very safe polymer into a very exciting piezoelectric polymer, which is basically the main focus of the talk today. And so the problem that we deal with here, specifically in this case, is to treat this very common disease called osteoarthritis. And so this is the disease that is manifested by the cartilage damage in the joint. And so when you have this disease, the cartilage, which is the buffering tissue between the bony ends, they get damaged. And then when you walk or you run or you're moving your joint, the bone, the two bones in the joint, they collide on each other, and they form a lot of this inflammation, the pain. And so this is a really common disease in the US, as you can see from the number, like millions of the adults get it every year. But the cure for this disease, it doesn't exist. Actually, there's no cure for osteoarthritis. But the main drugs like pain medicine or the anti-inflammation drug, it pretty much does alleviate the symptom, but they not really treat the disease. Now, to treat it, people need to regenerate the cartilage, and that's really challenging because this tissue doesn't have much blood vessel and all of that stuff. Is sitting in a very narrow joint space that is receiving very little nutrition. And so you can use stem cell therapy or you can use growth factor to regenerate the cartilage. But all of this had side effects. For example, like stem cell, it's very difficult to make sure that the cells stay there, and how to prevent them from the undesired differentiation into cancer cell or other dangerous cells. Growth factor is a biochemical cues, and so it carry a lot of side effects that you don't want to have a lot of them inside your body. And so from that limitations, when I started my job at UConn, I was gifted a book that from an orthopedic surgeon here, Dr. Kato Lorenzing, one of my mentors at UConn. And I read this book and I was really amazed by what being described in the book about the use of electrical stimulation on tissue regrowth. And so in this book, the author is Dr. Becker, described a very really amazing results from regenerate the whole limb from a rat that it had amputations in their limb. And the trick is that you have to implant a battery that it has two electrodes, and you're using that electrical charge or cues from the battery to promote the regrowth of bones, nerve, muscles, skin, pretty much everything, because the limb is everything. It's a complex tissues. And so from that early discovery, actually the field of electrical stimulation has been a while, and a lot of people working on this field for tissue regenerations. And in clinical setting, actually there's a real device used for electrical stimulation. And so this is one of them. It looked like a pacemaker, if you know what the pacemaker is. And you can implant it into the body, has a wire come out connected to some bone defects, or even in a spinal cord defect. And that electrical stimulation from the battery implanted here can promote the healing of the bone, and in the fracture or maybe in the spinal defect. However, you can see immediately this is really undesirable, really not favorable for implantations. The problem is you still rely on battery, right? It's toxic, it's non-degradable. At some point, you need to remove it from the body. And then you have wire come out, and that wire can be ruptured, can cause a lot of damage. And eventually you're gonna, it's not serving for the healing of the tissue because you're gonna remove it, and that damage the healing tissue. And so from that limitations, now adapting the traditional approach of tissue engineering, I was thinking about a way of what if we just using this approach by relying on the scaffold right here, but instead of using growth factor, using stem cell, now what if we can just integrate it with some electrical stimulation but in a simplest way, such that we don't have that wire, we don't have that battery, right? And if we can do that, we can have a really nice tissue scaffold that is self-power, self-charge, and we can avoid all of this problem in the in the electrical stimulation field as well as in the traditional tissue engineering field. And so that idea can be supported by the use of this material, so-called piezoelectric materials. And some of you probably know this, but some of you might not. So this material is called smart material. It's used extensively for industry, and I myself work on this material in my PhD. In one of the common materials, PZT, lead zirconium titanium. And so this is very highly efficient piezoelectric materials. What it means is when you apply a little force on it, it can produce a lot of electrical signal right here, and vice versa as well. You apply electrical signal onto it, it can deform and vibrate as you want from the control of the voltage. But the problem for the traditional piezoelectric materials is that they contains a lot of this toxic element like lead, and they're non-degradable. So you have to remove it again. And then, a lot of toxic effect that you don't want to implant this inside the body. And so with that, we come back to the surgical suture materials. And the question we're asking, what if you can transform this into piezoelectric materials so that we can have a safe, biodegradable, and at the same time, a self-charging materials that it can be used for this wonderful scaffold with inherent electrical stimulator in it. And so when I started my work at UConn, I started to look at the literature. And one thing I found is that this polyl-lactate polymer, which is a very common polymer used for a lot of tissue engineering scaffold, it actually can be piezoelectric. And so that came from the molecular structure of it. So if you look at the polymer chain, the CO double bond has a dipole, has polarizations. And so in the neutral stage, if you look from along the axis of the polymer chain, all of this dipole, they cancel out. But when you apply a shear force on it, for example, like the shear 2-3, which is a little bit mechanical engineering, but you can see that you can make them come off from the neutral stage, and you form a net dipole in the one direction. So that's so-called a shear piezoelectric. And so all you need to do is that you have to enhance that piezoelectric effect from single polymer chain to the whole macroscopic materials structure. And so to do that, you have to increase the anisotropic property of the material by forming crystals inside the materials and also align them in the same direction such that you can add up the dipole in each of the polymer chains. So you can do that by the traditional polymer processing like thermal stretching or drawing, but you can also do that with electrospinning. And so in this electrospinning setup, the key things that different from the traditional one is that you're gonna jet it on a rotator drum that is spin really quick. It spins really fast, and with the high speed of the this rotating drum, you can stretch the polymer further and align them better with a higher spin speed. And so here the SCM to showing you that more spin speed that you have more anisotropic property or more aligned nanofiber in the nanofiber mat. And so with that, we can create a piezoelectric nanofiber membrane, and we can control which side of it is positive, which side of it is negative under the mechanical activation, like in this case, the impact force of 20 Newton. And so with the material, we can form the tissue scaffold to perform this experiment in regenerating tissue like the cartilage in the osteoarthritis patients. And so this is the schematic to show you the vision of how this works. So you implant this cartilage scaffold, the PLA, piezoelectric scaffold, into the cartilage defect. And then you can activate it by mechanical force, maybe from exercise or maybe from something else like ultrasound, for example. And though the charges built up on the scaffold would be able to recruit the native cells, enhance the chondrogenesis as well, be able to regenerate the cartilage, which is usually very challenging to be regenerated. And so we form this 3D scaffold, as you can see on the screen. So we stack different layer PLA together with collagen so that we can form a little bit of 3D structure. It's not a fancy like 3D printing, but at least it gives us some thickness to be inserted into this major cartilage defect inside, the joints, for example, like the knee. And then we started to do experiment in vitro in which we're looking at how this piezoelectric stimulation can induce the chondrogenesis from stem cells. So we culture stem cell on top of our piezoelectric polymer, and we apply a mechanical force, but it actually, the value, it came from the estimated joint force that we have in the rabbit, that we do it later in the in vivo experiment. And so we induce this negative charge on the stem cell, and we're looking at the chondrogenesis, or the typical cartilage gene that is expressed by the stem cell. And we can see that the piezoelectric group with the force is always the group that has the highest chondrogenic genes expressions. And then from the protein, the ECM, or the extracellular matrix protein assessment, we also see that the piezo electrical stimulation is indeed induce a lot of this GAGs and collagen too, which is really useful for cartilage regenerations. And then we're moving to the in vivo experiment, in which we create this major cartilage defect inside the joint. So as you can see here, this is an image of the surgery in which we have a drill. We drill in a big hole inside the cartilage. So we form an osteochondral defect that is really big, such that it's not going to be able to heal if you just let it there. And then we train, and then we implant our piezoelectric scaffold into this defect, and we started to train the animal to hop on the treadmill. This is the stimulation that we're collecting the video from the animal hopping on the treadmill, and then we use some model to estimate the kinematic model to estimate the joy load that actually induced on our implanted piezoelectric scaffold. That's important because it would induce the charge. The mechanical activation is needed to induce the piezoelectrical charge that is driving the cartilage regenerations. So from that, we see from the exercise, we can see an amazing healing of the cartilage, especially after 2 months of the exercise. You can see the effect right here. It looks very similar to the native cartilage, the surrounding cartilage. While in the control group, either you don't have piezoelectric scaffold, you only have the normal PLA scaffold, or you have nothing else, no applied exercise. You can see that most of them just form this white tissue, which is mainly fibrosis tissue, and it never can be able to survive under the joint load or at load bearing for the load bearing function. We also see a lot of subchondral bone regenerations from this X-ray images, as you can see here. So the cartilage, the piezoelectric PLA, actually also can regenerate the bone, not only the cartilage, but also the subchondral bone, which is important to support this cartilage layer between the bony ends. And then histologically, we can see a lot of these GAGs proteins, right, from the suffering of staining, from the immunohistochemistry staining, we see a lot of collagen 2 expressed, as well as the H&E staining showing a very nice layer of cartilage. It completely regenerated on this load-bearing area, which is really a nice result that is rarely obtained if you don't have stem cell or growth factors. So in this case, we only use the synthetic PLA polymer that we transform it into the piezoelectric polymer. And so histological scoring also showing us, a very wonderful result in which the scoring for healing is much higher than the other control group. In terms of mechanical property, you can see that the cartilage that we heal, it really approaching the native cartilage for the mechanical properties. And so this is a very significant result because the cartilage need to be able to survive under the load. And finally, that's the image. That's a video to show you the rabbit after the implantation. The treatment was running really well without much pain and without much of this difficulty when running on the treadmill after the treatment. And for that, we're really happy. But one thing, as you can, if you notice, that we don't have a control over the mechanical activation. We train the rabbit to hop on the treadmill as a way to mechanically activate our piezoelectric scaffold. But we don't know how much actually the force that our piezoelectric scaffold is subjected to in vivo. And so this is also a very important question for the treatment of osteoarthritis, because as you know, this disease is a cartilage damage under the load. But people never know how much load that actually useful for cartilage regeneration. If you don't have any load, it's actually not good, because you need something to promote nutrition and cells and blood to go into the cartilage. But if you apply so much load, then it damages your cartilage. And so one of the things that we really want to do in our lab is to quantify the load that apply into the cartilage inside the knee. And so our effort, it actually turns out to be a couple of work that we try to develop this sensor, so-called a biodegradable force sensor. And it's also rely on exactly the same material that we make for the cartilage scaffold, which is a piezoelectric PLA. And so with using this material, we can develop this biodegradable sensor that when you implant it into the knee joint, you can measure the joint load, and over the course of time, you know how long it will last for. You can control that functional lifetime by control the encapsulation on this biodegradable sensor. And so here I just want to show you. So this is the work that we already published in PNAS 2018 and 20 about the sensor. It degrades over the time right here. And you can see that recently we developed a new sensor that is really accurate, and it can last very long time, at least like a couple of months from, this is a work that is unpublished, but technically you can see very accurately that the sensor giving you the same results. So the top is the commercial sensor that we implanted at the same time with this biodegradable sensor. This is just a dead animal. We are doing the in vivo animal right now with the live animal right now, but at least the dead animal showing you the function of the sensor, and it worked really well. So we're going to integrate this sensor with our scaffold, because they exactly make from the same material. And we hope that we can both getting some very exciting cartilage tissue that it both sends the joint load, it can wirelessly send that information to the outside, and we can use that information to tune and optimize the piezoelectric stimulation so that we can even enhance the healing outcome from this cartilage. And it not stop at the cartilage, but also other tissue like bone, nerve, muscle, which we also aim to develop the scaffold and the sensor for it. And so for that, you can see that we was able to show this wonderful healing from the piezoelectric scaffold. But the next step would be to test the scaffold in a large animal model, and then optimize the stimulation specifically, which we can tune the mechanical force such that we can get the best healing from our scaffold. So we're doing that, in the support of NIH and a lot of other funding agency. But one thing that we already did is that we developed another way to avoid one of the key problems in this implanted scaffold, which is the invasiveness of the implantations. So to avoid this invasiveness of implantations, now one thing we can do is we can make the scaffold into an injectable form. And so to do that, we would utilize some hydrogel. So in this case, we can somehow mix the piezoelectric nanofibers with some collagen hydrogel matrix. We can also do the injection without opening the knee and avoid that invasiveness from the surgery. And then maybe we can use some other mechanical activation like ultrasound to activate this nanofiber inside the hydrogel, right? Instead of using the load, which is mainly used for implanted solid scaffold. Now we can use it ultrasound to activate this single nanofiber inside the hydrogel. And so we did that by using exactly the same method. We first started with the membrane, with the nanofiber membrane. We make sure it's highly piezoelectric, and then we cryo-section that film into small nanofibers, and we mix it with the collagen hydrogels. So from that, we can inject it into the knee. So this is my student holding the hydrogel that she can inject to a small normal needle. And this is just a video to showing you that we can inject it into exactly, let me see, is that a video? No, this is here. So we can inject it directly into the cartilage defect under the guidance of the X-ray image. And so the big thing right here is the needles to, the tweezer to hold the kneecap, and then the syringe coming, the needle coming, injecting the hydrogel into the cartilage defects. So let me again. And so it's all done by the injection, and we don't need to cut and open the knee. And remember, this is not the hydrogel that to lubricate or to reduce the pain. We don't inject it into the joint space. We inject it directly into the large major cartilage defect to regenerate that cartilage, similar to a scaffold, but we do it in an injection instead of implantations. And so here there's some characterizations to showing you that the injectable nanofibers inside the hydrogel still perform the piezoelectric property as what we desire. Here's the output voltage that it can generate under the applied ultrasound activations. This PDLLA is not a control sample. It's a poly-D-L-lactic acid, and is because of the molecular structure is non-piezoelectric. No matter how you process it, and that's showing you here the no signal under the applied ultrasound. And then we can confirm that in vitro study, the hydrogel, after we chopping the nanofibers, the PLA nanofiber, and mix it with the hydrogel, we can see a highly chondrogenesis again from this sample, right, under the applied ultrasound compared to the control group. And so we're looking at how this piezoelectric material can do such enhanced chondrogenesis. And so this is just another result that we already published. But technically, the way it works is that you would generate this local charge in surrounding the nanofiber. So microscopically, it's almost neutral. But in each of the nanofiber inside the collagen, there's a local charge that when you activate it with the ultrasound, it can recruit the host cells as host stem cell. And then we can see that from the stem cell and the electrical stimulation, a lot of this endogenous growth factor like TGF-beta was actually released from the cell, and that induced the chondrogenesis. So we don't use any exogenous stem cell or growth factor in this case. So this is just the in vitro scratch test to showing the ability of the gel to recruit the stem cell. So you can see this group has a lot of cell migrate into it after the piezoelectric stimulations. And at the same time, we saw that if we use the inhibitor on the TGF-beta, we can see that the chondrogenesis effect that we saw in the previous slide actually suppress if you use this inhibitor on TGF-beta. So it's showing that TGF-beta is actually really good pathway that it help our nanofibers, the hydrogel, to induce the chondrogenesis. And so we again repeat the in vivo study in which we create the defect again. We inject the hydrogel into the defect. And so we activate it with the ultrasound, and you can see again that it can regenerate the cartilage very nicely, and it regenerates a lot of this hyaline cartilage, the collagen 2 cartilage, as you can see here from the histology study, that the staining of collagen 2 and suffering out always see the significant outcome from the last group right here, the piezoelectric with the stimulation, with the activation of ultrasound after 2 months of treatment. The result in the scoring of the histology image also showing the significant enhancement of the healing, right, from the pathologist who expert in the cartilage healing. And then the mechanical properties, again, it looks really nice, very close to the native cartilage. But one thing we notice is actually the hydrogel here can regenerate the collagen 2 cartilage, and looks really nice in terms of histology scoring, and the mechanical properties, but it's still a little bit less than what we saw in the implanted scaffold, maybe because of the charge, that it actually not as much as the charge that we can generate from the implanted scaffold. So we're still working on improving the efficacy of this injectable hydrogel. But the nice thing about this is that we avoid completely the need of the invasive implantation. So that actually is really a great improvement. And then not only the cartilage, we use this scaffold for a lot of different things. We use it for skin wound, as I showed you before, that they can even regenerate the whole limb, right, from the electrical stimulation. So somehow you still can regenerate the other tissue as well. So we use it to regenerate the skin wound. In this case, instead of negative charge, the positive charge is actually more helpful. And we use that surface to promote the activation of epithelial and fibroblast cells, which are the important cells for skin healing. But at the same time, another interesting thing is that you can utilize the negative charge to kill bacteria, and you can get this antibacterial effect from the piezoelectric membrane. So this is so-called a piezo catalyst. And actually, we published another work, which I'm going to show you later, that we can also use it to sterilize the membrane. And so this is really nice results, because for skin wound, the bacteria is a big problem, and how to heal it, but also how to prevent the infection is actually a key questions on the treatment of the chronic wound. And so we did the in vivo study in which we create this large wound in the skin of rat, and then we implant the scaffold. You can see that the piezoelectric stimulation group significantly heal the wound in a very short period of time compared to the other control, and even the positive control using Promogran, which is a commercial wound dressing that it has silver nanoparticles in it to kill the bacteria. And we can see that with the histology image, the piezoelectric scaffold actually can heal the skin very nicely, with a lot of this epidermis layer completely still regenerated. And we can even see some hair follicle reform, which is really amazing compared to the other control group. Even the Promogran is much less regeneration compared to the piezo stimulations. And so with that, I've been showing you, the use of this piezoelectric nanofiber as a scaffold, tissue scaffold, for a lot of different tissue regeneration, including cartilage, bone, skin, and it's actually we are exploring it for other tissue as well, but I don't have enough time and chance to report it right here. But besides that, we also use it for a lot of different things. For example, this is another, this kind of like a side project in which we're showing that we can actually utilize the same material to make this N95-like face mask, because we can utilize the piezoelectric effect to produce this electrical charge on the membrane, and that charge is very helpful to trap a small droplet of water or bacteria or viruses. And so here is some results that we published in Advanced Functional Material to showing you the advantages of this mask is that it's very stable, because especially a battery. So there's always a charge can be produced by the mask. When you breathe into it, or when you have a droplet of water impact onto the mass, you can keep producing that electrical charge. Meanwhile, if you use N95, you can see that you have a significant decline in the filtering efficiency for the small particle, like 1.0, like the particle that it has a diameter about 1 micrometer. So there's a significant decrease in the filtering efficiency if you keep using the N95 for like 2 hours in the humid environment. But in our case, the piezoelectric nanofiber filtering membrane has a really high stability. They have some loss in the filtering efficiency, but that nothing compared to what you have in the N95. So that is one significant advantage. But at the same time, you can also reuse it. You can sterilize it. As I say, that you can use ultrasound, you can even use autoclave and another method, just a microwave to sterilize this and kill the bacteria, and then you can reuse it. So this is just a prototype for the face mask that we make. The key thing here is that we want to make a biodegradable N95 face mask so that when you charge the mask into the landfill, it's gonna disappear after like 2 years or 3 years, and so that you don't have to worry about this huge burden on the environment by billions of these plastics that we trash every year by using the surgical and the N95 face masks. So that's another side project, but actually really exciting in terms of using the material, using it like a battery to produce the surface charge for a lot of applications. So that one side of the piezoelectric effect. Now, the other side, also very exciting to us, is when you apply the voltage onto the material, it also can vibrate and produce you something exciting like the acoustic wave. And so that is the other project. We work on the lab using the material as a biodegradable ultrasound transducer. And we use it for a blood-brain barrier application. And here is a recent paper that published in Science Advance. And so the work it comes from this crazy disease, brain cancer, glioblastoma. And the survival rate for a patient with brain cancer, glioblastoma, is very, very low. And because of that, any means we can do to improve the lifetime, expand the lifespan of the brain cancer patient, we would have to do that. So one key problem for treating this disease is that in the brain, there's a very well-known barrier, so-called a blood-brain barrier. And this prevents the drug to effectively go from the blood into the brain. So if the patient had a brain tumor, the medical doctor would do a surgery, remove the tumor, but the residual brain tumor actually is a problem. And to completely cure them, you need to continue to do chemotherapy, deliver the drug into the residual tumor. And the only way to do that is deliver the drug to the blood, because the blood can go to everywhere in the brain, inside the body, into the tumor site. However, that BBB, or the blood-brain barrier, prevent that drug from the blood go into the residual brain tumor. And so the most effective way so far that people found is that you can sonicate the brain by using ultrasound, and that allowed to open the this tight junction between the endothelial cells in the blood vessel, and allow the drug to penetrate into the brain transiently, and so that after a few 30 minutes or an hour, this pore can be closed up, and you have a nice barrier again without any damage on the brain, right? But the way to do the sonication is actually really problematic. We have to put a lot of arrays of ultrasound element around the brain, and we put the patient in MRI to monitor and make sure that the ultrasound intensity come from one of this array can be focused on this one single area at the solid tumor, because otherwise that high intensity ultrasound can destroy the other area of the brain, because they have to be used with a very high intensity to penetrate the skull or the bone that you have in the brain, right? And usually patient can only receive one MRI treatment, which lasts like the entire day to monitor this process. And so to deliver the chemotherapeutics into the brain repeatedly, and that very, very challenging, right? It's impossible to use this method from the outside ultrasound transducer. So recently, there's a way to do that. We can implant an ultrasound transducer right on top of the tumor, right? And from that implant, we can avoid the skull bone, and then we can just use a small intensity of ultrasound and focus that right into the tumor, and then you can repeatedly trigger the blood-brain barrier opening to deliver the chemotherapeutics into the residual cancer tumor that you have after the surgery to remove the solid tumor. However, this implanted ultrasound transducer, again, rely on the traditional piezoelectric materials, as I told you before. It's very toxic, it contains lead, and again, you need to remove it at some point, which is not favorable for the brain, especially if the brain is very sensitive and delicate structure. So for that, we was coming back to our piezoelectric materials, and we start to think about PLA, right? PLA is great. It has piezoelectric effect, but to make it into an ultrasound device, it has a big limitation in which the PLA piezoelectric coefficient is very small, nothing compared to the traditional ultrasound piezoelectric element like PCT, right? And so the problem that we deal with is how to create the same biodegradable, safe piezoelectric material, but which has a very high piezoelectric performance. And so recently, there's an exciting report on the use of glycine as a very highly piezoelectric materials. And so glycine is a common natural amino acid in our body, so it's definitely safe. It's biodegradable, right? And a lot of work showing that glycine crystal, so you have to make it into a crystal form because it needs to have some orientation of the atoms or molecules such that it can add up the dipole when you apply mechanical force on it. And so the glycine crystal is very highly piezoelectric. So that's good. It's safe, it's degradable. The only problem is very difficult to work with because it's really brittle, and you can see that it shatter like broken glass. Very difficult to make into ultrasound device because ultrasound need to vibrate, and so you need the softness from the piezoelectric element, right? And so to overcome this basically challenging problems to make the glycine crystal into a usable form, we think about a method in which we're going to make this highly piezoelectric glycine crystal, and then we're going to embed it into this nanofiber matrix, right? And we use PCl, another biodegradable and safe polymer, as a matrix, such that we can utilize the piezoelectric effects of glycine, but at the same time the flexible form of the nanofiber to make a very highly effective ultrasound element, right? And so here's the some fabrication process that we reported inside Advance. We grow the crystal, we grind it mechanically, break it into small nanoparticles. We electrospin it with the PCl, and we form this very, very flexible film that contain a lot of this crystal, piezoelectric crystal inside it. All right. And you can see that my student can bend, can twist, and can do a lot of things on this very soft membrane. So it allow the membrane to vibrate as well. And so we came to the device in which we fabricated similar to the sensor that I reported, I presented before. This is a very flexible ultrasound device, and we make it by encapsulate the glycine PCl piezoelectric nanofiber film with the two molybdenum electrode. Molybdenum is a metal that used very common in the medical implant, and it's biodegradable as well. And we encapsulate with another layer of neutral PLA, so the polylactic acid, but it's not piezoelectric, it's a neutral PLA for encapsulations. And so we can control the functional lifetime of this device, right, by controlling the encapsulation thickness. For example, here with a higher thickness, we can get the lifetime up to like 1 month or even more if you can change the molecular weight and the thickness of the encapsulations. So we do a lot of characterization to show we can control the piezoelectric output or the acoustic pressure that is generated from the nanofiber film. So less glycing, you have less signal, right? So the red, basically no glycing, it's just noise. And then when you apply the voltage with 1 megahertz frequency, you can get the waveform, the acoustic waveform that it does, the magnitude does increase when you increase the PCL. So here's just a video which I hope to show you how this works in real time. So this is a ultrasound generator, right? And so if you remove the word ultra, basically it's a sound generator. So it's like a speaker. So if you apply a voltage that has the frequency in the audible range, so you can hear, it actually can play some music for you. So here is a video to show you that. Hopefully you can hear the music. So we're gonna ramp up the input power. So the music gonna louder and louder. I hope you can hear the beat. So you can see that when the power, I stop it. Sorry, I'm gonna play it again. When the power is increasing, you can hear the beat. It's not a nice speaker because it's not meant to be a speaker. However, it can play music because it can vibrate and give you that acoustic wave or the sound wave. You can hear it if we can reduce the frequency. Yeah, I can hear it here. Hope you can hear it. You can hear that music from this biodegradable speaker. And then when that's a demonstration. When you make the encapsulation film thin enough, it actually can give you a really quick degradation. This is just a demonstration on that degradation process. So 2 minutes, then 5 minutes later, you can see it started to degrade. So this is pretty much like the first biodegradable speaker in the world. And you can see that it basically worked like the ultrasound device we implant into. Yeah, so my student pick it up, you can see that it's pretty much all gone. And so to implant it into the brain and use it for brain cancer treatment, one key thing that we really want to ask is how safe it is. And so we perform a really extensive long-term experiment on local and systemic toxic effect of this material. So we can compare the immune response in the local implant, to other traditional implant like stainless steel. And then we're looking at the systemic effect on different organs after the degradation of this device in the brain for 6 months. We monitor the animals, and we can see very nice safety profile from all of this assessment, and we feel really good about this device in terms of safety. And so we continue to do this in large animal model in the pig with the collaboration with John Hopkins right now in one of the NIH-funded grant that we have. But back into the efficacy of the device, we implant into the brain of the rat right here, and the rat here was induced with a tumor as well. And so we can show that we can stimulate the blood-brain opening, the barrier opening at a very deep area, like deep, like 2 millimeter deep, right, from the implant locations. And so you can see here, this is the result from this experiment, that if you looking at only the superficial layer, the PLA, which is our previous ultrasound piezoelectric device, we can open the blood-brain barrier at the superficial layer of the brain near the implant. But in the deep area, we only see the blood vessel, the red stained blood vessel. We don't see the green, which is the dextrin, the drug that we want to deliver. However, in our glycine and PCL device, we can see really a nice green that is showing the drug release from the device. And then in terms of the tumor treatment, you can see that in the initial day, when all of the rats getting the tumor, they all look the same. But over the course of time, only the rat that is treated with our device and the chemotherapeutics, in this case, of paclitaxel, which is very, very powerful chemotherapeutics, but it's not able to be used for brain cancer treatment because never can bypass this barrier due to the hydrophobic property of the paclitaxel. But you can see here with paclitaxel and our ultrasound device, we can reduce the tumor significantly. And then we can see that even when we take the brain out, the tumor size and also the brain looks really normal. And we can control the tumor very well compared to the other control group, even the group that using the paclitaxel without the ultrasound device. And then for that, we're looking at, again, different organs. We can see our device with the ultrasound sonication and paclitaxel give us a really nice organs without any systemic effect from the tumor. And we can increase the survival time of the animal like double from the use of the chemotherapeutics, the paclitaxel, right? So without our device, the drug basically useless. But with the device, this powerful chemotherapeutic drug can go into the brain and help us to kill the tumor and control that. So this is just a video to show you there's a small device implanted on top of the animal head right here. I hope you can see it. That's a wire come out. The animal is really healthy. It survived for a long time, and we can, whenever we want to deliver the chemotherapeutics, we can just connect the device into the brain and do the sonication again. So for that, I want to summarize what I've been talking about here. Pretty much my lab is biomaterial lab. We focus on this safe and biodegradable materials and use it for a lot of different applications, including vaccine, drug delivery, tissue engineering, and medical biodegradable electronic implant. So with that, I want to thank you for the attention and the acknowledgement slide for my funding agency, as well as the people that I collaborate with at UConn and other institutes. Happy to answer any question. I know that I'd run a little bit late out of the time, but happy to answer any question.
That's fine, sir. Thank you for the inspiring talk. It was very innovative. A couple questions came in. In arteriosclerosis, the majority of cartilage damage results from immune system activity. Have you observed any effects on immune cells in the PLLA, piezoelectric treated group?
Oh, wonderful questions. So the point here that we want to do is to regenerate the damaged cartilage, and that's basically the origin of this osteoarthritis disease. But yes, you're correct that in OA, because of the loss of the cartilage, the bone collide on the bone, and that actually form a lot of inflammation, and immune cells basically recruited into the site, the injury site, and started to destroy other tissues surrounding it. But in this case, we mainly focus on the regeneration or the regenerative property of our piezoelectric scaffold. We didn't look at much in terms of inflammation, but from the in vivo data that we saw, it actually there's not much inflammation as well from the cartilage defect that we treat with the piezoelectric scaffold. In vitro, we do see that the piezoelectric stimulation actually can also help to reduce the inflammation. We had that experiment done. We didn't publish it, but we did see that. However, in vivo, we really don't focus on that. We focus on the regenerative property of the scaffold. And it seems like when we can regenerate the lost cartilage, and when the scaffold here recruit the native stem cell, especially from the subchondral bone or from the synovial fluid, we see that stem cell, when it move in, it actually help us a lot to reduce the inflammation. and that actually is a really good thing for the treatment of this disease. Thank you. Any question? Any other question?
Thank you. Yes. Can you please explain the mechanism of sequential release of drug 1, 2, 3 from the microneedle? How are you accommodating the 3 drugs?
Oh, yeah, sure. So in the vaccine, I guess you're asking about the vaccine
Yes.
delivery. Basically, the microneedles here are made of 3 different types of the patch carries 3 different types of microneedles, and each of them would have a different PLGA shell in this core cell structure. And so with different PLGA shell, you have a different degradation time. And so with the different degradation times, you have the drug come out at a different period, right? So I can have PLGA, make out of low molecular weight and high molecular weight and more lactic, right, or less glycolic components, such that I can control the degradation rate of the polymer shell, and that dictates how fast or how slow the vaccine release from this core cell microneedles. And so we put 3 of them into the patch, into the skin. 3 of them release at 3 different times, so that avoid the need of booster shot or booster injection. One of the injection just replaced by this patch.
So it's a
Does it?
time release. You don't control the release time or duration. Whenever it dissolves, it releases.
Right. So we control it by controlling the PLGA shell right here, and we have a calibration to make sure that at what time this PLGA is going to degrade and the drug going to release out from that. So I didn't show here the release profile, but we should have a release profile that is show a very sharp and release of the vaccine. Let me see if I have it here. I don't have it here. But you should have a sharp release from the core cell microneedles.
Thank you. How did you evaluate the piezoelectric effect of your materials in wet conditions? Because most of the physiological conditions are wet.
Correct. Yes. Yeah. So we characterize this as the as a normal way we characterize the other piezoelectric materials. So we would apply the mechanical force onto the materials, and we measure the output piezoelectric charge until we can vary the force and measure the output charge. So that quantify the piezoelectric performance of the material, right? So how much output voltage or charge versus the input mechanical force. However, you're right. In the physiological environment or in wet environment, when you had a lot of this conductive ions flowing the in the body fluid, uh, your charge going to be green, right? However, the charging effect is still helpful, right? So it's similar to when you have an electrode that you put it into a solution. When you have the charge on the electrode, the cell, the protein, all of that would be able to see it, right? But it's not going to stay there for a long time. It only stay there for a short period. And after that, the conductive environment would neutralize the charge on your surface. However, that little charge and that short period when you have the charge that it comes from your implant scaffold is already useful for recruiting cells and attracting the protein, the ECM protein, and that actually helpful for tissue regeneration already. So you don't need to have the charge to stay there for a long time. But the short period of time already useful. And remember, this is a piezoelectric material, so the charge come from the internal atoms reorientations. So on the other words, the charges come from the inside of the material, right? So it's not like the surface charge that you have like in the N95. You have a surface charge at the beginning and then it get neutralized. In this case, the piezoelectric can produce the charge whenever you mechanically activate it. So you always can have that charge when you activate the material.
Thank you. Thank you. For what duration should the patient utilize ultrasound on their head? Furthermore, is it not considered targeted therapy?
So, okay, so the first part of the question is how long the ultrasound should be used for. We haven't optimized the mechanical activation, so we don't know. Maybe from what we saw is the 2 months stimulation is better than 1 month. But what about 3 months? What about instead of doing every day, we do it once every 2 days, stuff like that. It hasn't been optimized and studied yet. So I cannot say which one would be the best. But at least the ultrasound stimulation and the exercise are the very non-invasive and easy exercise and activation that we can have for activating our piezoelectric scaffold. So we use that for now, and the parameter that we establish is good enough to show the good regeneration. But optimization is another story. We haven't done it, so we cannot say in what duration of the ultrasound would be the best. We only know that there's some duration we already used that it worked to regenerate the cartilage. And so the second part of the question, what is the second part of the question? Can you
So
remind me?
on the first one, do you use like 5 minutes a day, 10 minutes a day,
We use
2 hours a
20
day?
minutes a day. We use 20 minutes a day, and we do every 5 minutes. We stop for like 3 minutes, and then every 5 minutes. So we do 5 days a week, 20 minutes a day, and we do like 1 month or 2 months. We rest the rabbit after the surgery to inject or to implant the scaffold. We rest it for a month before we do the ultrasound or the exercise activation.
Okay. And the second part says, is it considered targeted therapy?
I don't understand what you mean by saying targeting therapy.
Like you're targeting the BBB opening,
Oh, that's another. Oh, I see. So yes, so for the ultrasound application in terms of the BBB opening, yes, we implanted the ultrasound device right on top of the brain cancer, of the brain tumor, such that the ultrasound intensity does focus. It fixes the location is fixed, and it does focus exactly at the location of the tumor. So we make sure that whenever we turn on the ultrasound, the area where the tumor is would be the area that receives the most intensity.
Thank you. A lot of other questions came in. What duration should what are the dimensions of the microneedles? How can the fabrication process be controlled to achieve a layer by layer structure? Which technique did you use?
Yeah, so the technique that I use, so basically I adapted from the mass aligner process in the microelectronic fabrication. So if you're working in the clean room for making electronic chip, a very common device that it use is the mass aligner. And the mass aligner basically it stack the one layer of the electronic chip with another layer, for example, the layer of electrode on top of the layer of insulator and on top of the layer of semiconductor. So I used that mass aligner device. I modified a little bit. I put in some controller on the temperature so that when I align them and put this layer together, I can form, I can basically seal them together and form this microneedle structure as you see here. So this is a shell, and it basically capped by another polymer, by another PLJ layer, and then there's a supporting array that we also align and put them together to form the microneedles. I hope that I answered fully the question. I don't remember the other part of the question.
Yes, you did. Yes, you did. And I
Okay.
think, in the interest of time, I'm going to go through the other questions. If people have
Okay.
extra questions, they can email you, I think. Regarding the mRNA stability, at what temperature is the vaccine considered stable?
Oh, regarding to that mRNA, which I show briefly here, the work that we did with mRNA. So we use 37 degrees C because in our microneedles, we need to implant them or we need to insert them into the skin. And the skin temperature usually 37 degrees C. And we want to embed them into the skin for like a year. So we have to stabilize them for a year. And this is the temperature that we use. We also use a very high temperature as an accelerated temperature to screen some sort of good formulation for stabilizing the mRNA before we do this long-term study. But usually we use 37 to 45 degrees C as a temperature to target.
Thank you. If you want to use the material for applications that the mechanical moment is not involved very much, how would the piezoelectric material can provide advantages compared to the conductive polymers?
So first of all, the piezoelectric materials here were technically PLA. There's nothing else, right? So if you think about that, polylactic acid is extremely common material and safe for medical implants. So we affirm that it's safe. That's the first thing we want to do because we don't want to go through this crazy safety study and get FDA rejection. So first of all, the safety, we feel really confident and feel really good about the material, right? Conducting polymer sometimes have something, in terms of degradation, right? It needs to be degraded as well over the course of time. We want to make the tissue scaffold degradable. So the degradation byproduct from the conducting polymer or from others, really exotic polymer could be an issue for FDA and for the regulation as well as for translational approach. So because of that, the PLA really good in terms of like affirming that we have a good safety effect. Now, in terms of the electrical stimulation, this is piezoelectric, right? So what it means is like a battery. So when you apply the mechanical activation, not only the exercise, the joint load, but you can see that we can also use something like ultrasound, such that the patient doesn't need to move at all. The ultrasound can just apply when the patient sit still or lie on the bed, and that ultrasound mechanically activate, sending the acoustic wave and pressure to activate the piezoelectric scaffold. And so we can avoid that movement of patient and just activate it. We can use this as a battery, a self-power battery, right? So this is much nicer than the conducting polymer because conducting polymer, to do the electrical stimulation, you need to have a battery. You need to have something to sustain that electrical charge, right, on the conducting polymer. In this case, the polymer, our piezoelectric polymer, produce that charge every single time you activate it. So it's a self power and self charge tissue scaffold, which I don't think conducting polymer can do that.
Thank you. Piezoelectric with ultrasound for cartilage regeneration, giving ultrasound to stimulate the material. Does it affect the normal surrounding tissue?
Well, we use the ultrasound intensity that is always less than 0.5 watt per centimeter square, and that's the threshold for low intensity ultrasound use in clinic, and that makes sure that the ultrasound intensity not going to damage any healthy tissue. And the frequency here, sometimes we use 40 kilohertz, sometimes we use 1 megahertz, but the intensity always make sure that it low intensity such that we avoid the heating effect, we avoid damage on the other healthy tissue. So I don't think that there's any concern on the other healthy tissue.
Thank you. In the case of PLLA short fiber with collagen, does the load exercise only will provide enough fuse to regenerate cartilage?
I'm sorry. So can you repeat the last part of it?
Yeah, it's a good question. Does the loading and exercise, does it provide enough cues to regenerate the cartilage?
Yeah, yeah. So we only need a little bit of charge. And so mechanical activation doesn't need to be much to activate that piezoelectrical charge on our scaffold. And actually, if you produce so much charge, it can create a very toxic effect. The charge can basically create the ROS, the reactive oxygen species, and it can cause a crazy thing in the body. So you only need a little bit of charge. And this is what is nice about PLA, that it only produces a little bit of, it has a very low piezoelectric performance, so it produces a little bit of charge, but not enough to promote the tissue healing. And usually that charge we quantify in vitro in the solution is about like 100 to 500 picocoulomb on a 1 centimeter square of the material.
Thank you. Also, maybe a simple question. Why do we need external stimulation ultrasound for the hydrogel?
We need it because the hydrogel different, right? Because the fiber is basically embedded inside a soft matrix. So if you can induce the mechanical force by the load, right, by mechanical impact or loading, like a joint exercise, for example, but that would apply on a soft hydrogel, and inside the soft hydrogel there's nanofiber. So that nanofiber wouldn't see much mechanical activation, right? In the meanwhile, if you apply the ultrasound, hydrogel can catch and pick up the ultrasound very well, and that vibration would make the fiber inside the hydrogel vibrate really nicely and produce more charge than if you apply the mechanical loading onto the hydrogel. So that is why we use ultrasound. But at the same time, it's a nice way because, like the other questions say, the patient can just sit and lie on the bed. They don't need to do exercise, especially at the early phase of the cartilage damage, because it's very difficult to train them on the treadmill, right? Or to moving the joint. Now you can just apply ultrasound and passively let them passively, sit or lie on the bed, and you can heal the cartilage from that.
Thank you. What is the molecular mechanism of piezo bioelectricity? The electric field generated by the material creates gradients, and how does this affect the regeneration?
So if I understand the question correctly, that's the molecular mechanism of piezoelectric effect. Is that correct?
Yes.
Okay. So I briefly talk about that. So it comes from this atom, this basically Cl double bond in the PLA, right? So this is a dipole that in the neutral state, this dipole is basically cancel out. So there's nothing, no electrical output. But if you apply the shear stress, so you can think about this, you can twist this, go down, and all, these two guys go up, and that, like this image, and that form a net dipole in a polymer chain, right? So under the shear force, actually, KLA polymer, one single chain can produce the piezoelectric effect, right? The problem is that you need to add up all of this dipole in one single chain to form a net polarization on the entire material. And that can be only done if you make the material into an anisotropic form. On the other words, that you want the align this polymer chain in the same direction so that you can add up that dipole, right? So to do that, you can put the polymer into an oven, for example, and stretch it under the high heat that form piezoelectric PLA. Or you can do electrospinning, as I show you, with the really um, uh, rapid rotating collector, such that you can stretch and align this nanofiber in the microscopic scale as well. So that how this can be a piezoelectric, the PLA can be a piezoelectric.
Thank you. Great presentation. How can you precisely control the ultrasound pulse and energy of glycine film for disrupting the BBB. Are these piezo materials compatible with cell culture?
Yes, we did. So how to make sure, how to control the intensity um, from the glycine. So the first thing we would need to do is we have to we have to calibrate it in the lab. So we have to put it in a beaker that containing water, and we have to have a hydrophone, which is a device to measure the acoustic wave intensity. And we're just moving it, in front of the ultrasound transducer, make out the glycine. And then we measure the intensity in front of the device, and we would know at what point in the space that intensity can be focused on, and what intensity, what magnitude. And so we can tune the voltage that we apply into the device to tune the acoustic wave in the output, right? So it proportional, it's linearly proportional between the output acoustic wave and the applied voltage. So you can tune the applied voltage to tune that intensity. So that's the way that we control it. I don't remember the second part of the question.
Are these piezo materials biocompatible with cell culture?
Yes. As I said, not only the cell culture, we did the cell culture, we published that data as well, but we also did extensive in vivo study for the toxic effect of this biodegradable ultrasound transducer in the brain. And you can see here from the result that it's extremely biodegradable and safe.
Thank you so much. That's all the questions I have. I appreciate your time and the fantastic talk, Professor.
Wonderful. Thank you so much. Great question. And again, happy to be here and interact with the wonderful people in the Institute.
Take care. Bye-bye.
Yeah, thank you. Bye-bye.